Thank you very much. It's a pleasure to be here. I'm sorry to say my first time to Oxford, but it's been very enjoyable so far. So I'll talk a little bit about my research and kind of general big data ideas I've been pondering, and as Guy said, feel free to interrupt me at any time. So a bit of an outline. First, I'll kind of give you a brief overview of big data, or what I think big data is, why bother, and then walk you through 3 applications, or more like 2.5. I just wanted to talk a little bit about adolescent cognitive development, a topic I'm quite interested in, how to integrate explanatory levels to understand aging, so how to make most use of these large data sets we have, and then finally dipping a toe into big data with capitals. So what is big data? I think the best definition is by Dan Ariely. And I think this captures most people's slight sense of unease and kind of excitement about it. And I think this also kind of captures that we're all trying to figure out what to do and how to make best use of this new thing we have. So does size matter? Well, there's this quite nice paper that was published last year on big data. And they basically looked at data input rates and processing rates. And basically the kind of winners ultimately were genetic analyses in terms of complexity of doing the data analysis, but astronomical data for data input rate. So this is the Australian square kilometre array, which is essentially a big camera. And who wants to hazard a guess at the data input rate for the square kilometre array?
Terabytes per second.
How many?
Terabytes? I don't know how many.
Pretty good. 750 terabytes per second is going to be the future rate. So if you think your data is big, maybe not so much. I'm well aware, I'm very much dating myself by a Crocodile Dundee reference here, but I took my chances. Yeah, imagine being these kind of system administrator there and kind of going home over the weekend. It's like, oh God, another few zettabytes to deal with on Monday morning. So that's big, objectively big. So you might say, well, do I need these big data sets? So who here has thought this out loud or to themselves? Right? It's a perfectly sensible thing to think. Like, well, if I found it here with my kind of crappy NS14 sample, then surely there must be something there. Well, this is a paper by Andrew Gelman that I urge you to read, but not before sleeping, because it includes the most terrifying plot in all of psychology, which is this one. So they basically summarize their point by, this is what 6% power looks like. 6% power means if you find an effect, the effect size you have is at least 9 times overestimated. So what you often see in small studies say, well, yes, we only study 10, 20 people, but our effect sizes comes at 1.5, so that's fine. So now 10 is enough. That's not true. You're getting an overestimate of the effect size. But far more terrifying, and if you sleep tonight, I haven't done my job, is what they call the type S error probability, which means if you find an effect in a small sample and it's significant, there's a 1 in 4 chance it's actually in the wrong direction. Now, if that doesn't scare the hell out of you, then I don't know what will. So never, ever have that thought again. And if you think, well, but who has 6% power? Surely nobody does. So there's this paper by Kate Button a few years ago who looked at imaging analysis, and it's mainly EEG. They found that 31% of papers have power less than 11%, which is firmly in the, oh God, how many of our effect sizes are in the wrong directions? And these include drug studies. You might be concluding it makes someone better, and actually it makes someone worse. So, are we going to fix all those problems by just having bigger data? Well, my favorite quote about big data is actually this by Overleeuw, which I think captures the naive approach to big data quite well. It's like, well, all we need to do is just have more data, and then suddenly the heavens will open and insight will come to us from above, and we'll know what to do and why. Clearly, the best you can say about big data is that it's a necessary but certainly not sufficient condition for doing good science, or at least big enough data that you're sufficiently well powered. So my favorite definition of big data comes from Tal Yarkoni, who summarized it: the minimum amount of data required to make one's peers uncomfortable with the size of one's data. I think that actually, I'd say quite funny, but it's also captures something quite true, because suddenly you're out of the realm of the way you used to do science, and that helps doing science in a new way in a lot of ways. So what do I think big data is useful? It's like, well, when it inspires new questions, and that also means feeling slightly uncomfortable with the size of your data set, suddenly you might start to think about new questions. To look beyond your current domain, so whether you're a psychologist who's now currently suddenly looking at genetics, or suddenly thinking, well, is this effect different by country, because suddenly you have data sets for multiple countries. To develop and apply new methods, so I find in my research, the larger data set you get, or the richer data set you get, suddenly you start to think about new questions, and they often need new methods, and sometimes those methods haven't been developed yet. So then you ask a bunch of statisticians, say, does this exist yet? And sometimes the answer is no. And my background is that I'll argue and give you at least two examples, is that it's, at least in principle, possible to test theories with multivariate data methods. So you can translate some theoretical conception you have by integrating all of the data rather than simple bivariate kind of t-tests and correlations, and see if you can represent a hypothesis you have as a full multivariate theory. So that's the why. What is big data and why bother? So I'll walk you through two empirical examples I've been working on, and I'll focus on two parts of the lifespan that interest me, the kind of rise and fall of executive functions or higher cognitive abilities. So the first sample will be in NSPN, this Neuroscience and Psychiatry Consortium, data from Cambridge and London. And then secondly, the CAMCAN data set we've been working on for the last few years. So the question that interests me a lot is how and why cognitive abilities develop reasonably rapidly during adolescence and early childhood. And it centers on this kind of the positive manifold, which I would argue is the most replicable and robust finding in all of psychology, which is the fact that scores on ability tests, almost regardless of their content, are generally positively correlated in the population. So that's been established for at least 100 to 110 years. And one of the famous explanations of this by Spearman was to posit this G model for this general intelligence. So people have a G of different sizes or strengths, and that explains why some people do better across the board and other people do slightly worse across the board. And he called it mental energy. So there's a single thing you have that you use for all kinds of tests. And I think it's quite important because we know that G-scores predict health and morbidity, income, education, and even levels of psychopathology across the lifespan. And it's surprisingly robust. This is amazing data set from Edinburgh. They found the test scores from this cohort from the 20s, and basically they found almost all the people who were still alive and got them back and did the same test 70, 80 years ago. And the correlation of some of these tests is 0.6, 0.7. So 7 decades later, you can still do a decent job of predicting people's test scores. So it's A, predictive of lots of things, and B, surprisingly robust. So I want to know where it comes from and what its underlying mechanisms are. The interesting thing is that development has generally not been on the agenda of kind of individual differences in psychology, at least not as much as I would argue it should be. There's a famous book called The G Factor by Arthur Jensen from the late 90s, which is a solid 800 pages of kind of graphs like this, making the case for this G factor, but the word development isn't even in there, and it doesn't address age-related changes at all. Well, why is that a problem? Well, because if you think of the G model, well, this is at least in principle, a plausible parsimonious explanation of why some people do better across the board. But there's this other competing model. So there's one that Thompson already in 1916 proposed the Bonds model, and basically what he said, well, we can also make the assumption that there is no G, but there are lots of independent cognitive abilities, and whenever we develop a test, like test B or test A or test C, they sample a subset of these abilities. Your ability to see or to write or to kind of memorize. And because of our imperfections as researchers, we're always going to sample overlapping subsets of these, in principle, uncorrelated cognitive abilities. And basically, because of the nature of our test construction, you get this area of overlap, ABC, which has nothing to do with the cognitive architecture of the mind, but just our incompetence as scientists, which means the world will look like there's a single ability. So he didn't necessarily say he believed this was the case, but he showed mathematically that the data looked exactly the same. And there's this rather nice new model called the mutualism model, and I'll go to in a bit more detail on that. That argues, well, if the only assumption you make is that, say, there are 5 fundamental cognitive abilities, and they have positive interactions with each other, so they develop or they help each other develop, the interesting thing is these three radically different models predict exactly the same covariance matrix. So you can get 1,000 people with this matrix, or 1 billion. That's not going to help you differentiate between these three completely different models. This is a case where big data is nice, but it's not going to solve this particular conundrum. So what will? Well, if you think about development, by the way, this is the first hit you get if you Google Spearman, and I stuck with it. So the Spearman G model posits a single underlying ability. So we can think of, well, even though he didn't really address development explicitly, we can think, well, what prediction falls naturally that explains why people get better? Well, when you have a small g when you're young, and that grows into a bigger g, stronger, more pronounced, higher. So that's what explains cognitive development. It's a relatively simplistic model, but it might account for development. What about another theory? So this theory is called investment theory that focuses on kind of two domains. It says, well, there's in the general intelligence, there's kind of two subdisciplines or two subspecialties. So the first is fluid intelligence, and I'll get into that in a bit more detail. So fluid abilities are kind of general problem-solving abilities that don't depend on any knowledge. So it's abstract on-the-spot reasoning of the types of fill out this kind of what matrix is next. I'm sure you've seen those types of puzzles. And the other type of cognitive domain or intellectual domain is called crystallized knowledge. So it's essentially the sum of all your knowledge that is kind of accumulated across the lifespan. So this includes vocabulary, but also knowledge of geography, etc., etc. And Cattell made this interesting claim. He said, well, he called this investment theory. So he said, you invest your abstract reasoning ability, your kind of general problem-solving ability, to gain crystallized abilities across the lifespan. So what does this mean for our kind of theories of development? So he argued, well, this year's crystallized ability, so if I test you now, that basically means you invested your fluid abilities across a certain period. And if we think of development, then that means that, say, the kids who start out with a little bit more fluid abilities initially have a little bit more crystallized abilities, but because they have more to invest, they manage to acquire more across the lifespan. So these fan out across the lifespan. And it also explains the fact that although fluid abilities start to decline quite quickly, depressingly from your mid to late 20s, crystallized abilities can improve until your 70s and even 80s in some individuals. So this gives us a different kind of prediction of what we would expect to see if we follow people longitudinally over the lifespan. And finally, this mutualism model I discussed earlier is actually stolen from ecology, where they had the same problem where, well, you can think of, say, the health of an ecosystem, of a pond or a forest or whatever it may be. You can summarize the health of that ecosystem in a single number. It's either doing well or it's not doing well. But of course, nobody thinks that that means there is this underlying kind of health gene, so to speak, of this ecosystem. The idea in ecology is that you have these multiple interacting kind of subparts of the system, such as the fish and the algae and the plants, etc., and they kind of balance each other out and feed on each other and interact with each other. And you can kind of think of cognition or cognitive development as a complex ecosystem in the same way, in the sense that if you just make the assumption that, say, your vocabulary is facilitated by your working memory, which is a totally plausible assumption, Van der Maas showed, quite surprisingly perhaps, you can start out with uncorrelated abilities in very young children. The only assumption you make is when they develop, whatever their starting point is, there are positive interactions. And he showed you end up with one big factor where everything is correlated positively. So you can summarize, much like the health of an ecosystem, with a single number pretty accurately, but that doesn't mean there is anything latent or underlying all these domains. So how are we going to translate these theories into models? How can we see which one is right in our data set? So I'll give you a 30-second primer on the types of models I've been using to address these questions, called structural equation modeling. So it's quite a powerful multivariate method, and the basic idea is you take some causal hypothesis you have about the world, and you translate it into a certain model, and then you see whether your data is compatible with your causal prediction. Now, of course, that doesn't mean we can use it as an easy causal discovery mechanism, but we can see whether our data is compatible with a set of predictions we make. So what do these look like? You'll see these kind of almost butterfly net looking plots. But the basic idea is you have squares in an SEM plot are observed variables, so they would be, say, the average reaction time on a certain test or some score on an ability test. Circles are latent variables, so they're these things like your mythical G, so this underlying ability that explains your performance on the observed variables. Directed relationships are things like factor loadings and regressions. So that posits a causal hypothesis. So for instance, you have a G and that causes your score on all these tests. And then finally, this undirected relationship, such as covariances. Now this is very minimal, but just to give you a bit of a framework when I start showing you some of the thoughts of what you should be thinking about. So this specific type of model I've been using recently, because I think it's quite a rich way to look at developmental data, are called latent change score models. And I'll walk you through why they're useful and why I think you might be interested in using them. So say we have, the simplest case, we have one score on 2 times, time 1 and time 2. Normally you do something like a pair t-test, right? So you have an average on y1, an average on y2, and the second occasion you just see do people go up or go down. The nice thing of a latent change score model is that you posit this latent change score here. So these are essentially your scores on the latent change factor. And you specify that by regressing time 2 on time 1. So essentially this becomes the residual. So if you go up, they're positive. If you go down, they're negative. Essentially this model is equivalent to a pair t-test in the simplest case, but you get 2 extra parameters for free that I think are quite interesting. So firstly, you get the covariance for free, which means does the degree of change depend on where you start? So the types of cases where that almost always matters are things like depression interventions. So the ones who benefit most from an intervention are generally the ones who suffer most. And the second parameter you get for free is the variance in change. So does everyone just gain 1 point, which would be quite uninteresting, or is there individual variability in the rate of change between time 1 and time 2? So this is a really simple model with just 2 observed data points, and yet you get a richness that I don't think you get from a pair t-test. So you can distinguish these cases. So if you just did a pair t-test, so on time 1 and time 2, all of these 4 scenarios here would tell you, yes, there's improvement between time 1 and time 2. But I hope you agree that these are quite different. Here, this kind of uninteresting everyone gains a point. Here is the most realistic case where there is on average gain, but some people decline. Here you get the kind of depression case where people who are most depressed are going to gain most, and the ones who aren't really depressed are going to benefit least from some intervention. And this is a change in the positive covariance. So it's a relatively easy way to tease apart these quite kind of developmentally, interestingly different scenarios. So deep breath, we'll take it one step further to the bivariate case. So we can now have 2 domains, y2 and y1. And we're going to model the rates of change on both domains. So now the cool thing is we can predict the rate of change on each domain as a function of itself, so the self-feedback parameter. So remember, in the case of depression, this would be negative because the ones who start out most depressed are going to benefit most, and the ones who start out least depressed are going to benefit least. But we can also model these cross-domain coupling parameters, so the blue parameters. So that basically means does how much you improve over time on score y2 depend on where you start in the other domain. So if you want to take like an athletics metaphor, it would be something like your rate of increase in muscle strength depends on your stamina time 1. Because if you have a lot of stamina, you can do a lot of repetitions. So that doesn't mean stamina and muscle strength are the same thing, but there is this coupling here between how much you can, kind of the preconditions for development over time. Mathematically, this looks like this. So it gives you 2 parameters, the self-feedback parameter and the coupling parameter. And for our interest, the coupling parameters are most interesting. So to get to the data, this is this neuroscience and psychiatry network, where we have about 800 kids measured on 2 occasions. We have vocabulary, which is the WASI vocabulary, which is essentially things like name pictures displayed in a stimulus book or give definitions of words. And the other domain is matrix reasoning. So as I mentioned before, these kind of abstract reasoning puzzles. Nice psychometric properties has been used for a long time, so kind of trust them more or less. So what does the data look like? Well, this is your matrix reasoning data. So here you need the eye of faith to kind of squint a bit and see that there's some development. There are probably some individual differences. And the vocabulary data, something similar. So now we want to make sense of both these domains at the same time. So if we go back to our latent change score models and the theoretical accounts we had before of development, we can have something like G-factor theory, which would posit a G-score at time 1 and time 2, where what we're interested in is did your G grow, and does this model account for what happens in our data. The second model is this investment idea, right? So we have our matrix reasoning test, which is our fluid ability test. And the investment theory basically says your degree of change in vocabulary should depend on how much you start out with in terms of abstract reasoning ability. So this is like the engine that drives your growth in knowledge of the world. So we only need a single parameter. So what I quite like is that you take this kind of plausible theory full of words, and then you translate it into a model that can be right or wrong. And then finally we have this idea of mutualism, which posits no, all of these cognitive domains, or at least quite a few of them, must have positive interactions over time. So here we have the investment theory, but we also have this reciprocal path, namely, if mutualism holds, then we should also expect greater gains in fluid abilities from those who start out high in vocabulary. And the nice thing is we can then fit these models to the data and compare them. So they make three slightly different predictions of what their data should look like. And we then use yeah?
Just go back to one more.
Yeah.
That double-headed arrow on the left of the mutualism.
This one?
No, in the mutualism,
Yeah.
the curved arrow that's right on the left.
Yes.
Because that represents the fact that there's a correlation between matrices and vocab right at the start. Yeah?
Yes. It's because, yeah, you're totally right, because we only have a snapshot of development that's arguably pretty late. So these are 14 to 24-year-olds. Ideally, you would start them aged crawling around on some task. But we know that they're going to be correlated because even if mutualism is true, we would expect the scores to already be correlated at age 14. Yeah, you're totally right. So we assume we allow them to be correlated at time 1.
So do you have to start off from a positive significant correlation there?
Not necessarily, no. The mutualism model, I mean, it could be the case that they're uncorrelated. If hypothetically all development is explained by the mutualism model and you tested kids age 1, it might be that they're uncorrelated. But that's essentially something you can test empirically whether that's the case. But because we think mutualism, if it happens, it definitely also happens before age 14, we allow them to be correlated at the start. So we can then use the framework of SEM model comparison, where I'll spare you the soporific details, but tell you that the mutualism model in this particular data set fit by far the best. So even though it has a degree of freedom less, so it's slightly more complicated, slightly more arrows, it still outperformed the other two competitors quite considerably. So the mutualism model seems to be the best account of our data, which is a relatively simple data set. It's only 2 measures, only 2 occasions. But we do have a principled way of saying, well, yeah.
Well, just a really small question, but would you need a much larger sample to test the mutualism? I think it would look like you would need to estimate quite a lot more parameters, or is it?
So it's only 1 parameter more than the G-factor model. Yeah.
Oh.
It's not too bad. And the sample size you need depends on your expected effect size. But we have about 800 here. But yeah, you're right. That, I mean, much like things like moderations and kind of mediations that people are quite interested in, generally it's nice to have at least 500 people to have a decent chance of detecting a small effect. So on this basis, the mutualism model did best, but of course, model comparison is the only thing we're interested in. So we can now look a little bit at the parameters in a bit more detail. So I'll walk you through them, what the parameters are, and the suggested level of interest you display. So the basics are relatively uninteresting, which is the covariance at time 1 that you rightly pointed out. So we expect them, or at least allow them to be correlated, because we don't expect them to be uncorrelated. And because we have kids in the first wave ranging from age 14 to 24, we also have a covariate of age here, because they didn't all start at the same age, so we need to remove that to be able to account for development. So perhaps unsurprisingly, A's are correlated at time 1, and B, age predicts performance. A bit more interested are the dynamics of change, which include the change intercept, the change variance, and change covariance. So what does that mean? It means they improved between time 1 and time 2. It means there were quite considerable individual differences in the rate of growth. So in other words, if I fix this parameter to 0 and then test my model again, it'll collapse and fail miserably. So there are individual differences in rates of growth. And finally, the rate of change is correlated even when I account for these regression parameters. So that means I haven't fully explained away the fact that these changes in each of these domains are correlated, which isn't too surprising because we only look at two tests that are simplistic representations of two single domains. So the most interesting effects are the mechanics of change, so the self-feedback effect. So often these are almost always negative because they also capture things like regression to the mean and the age-related dampening. So you don't develop linearly into your late 20s, so you start to slow down. So if you start to slow down, there's essentially kind of a developmental ceiling effect, so that'll appear as a negative self-feedback parameter. But interestingly, here you see the two parameters that represent the mutualism model, or the idea of mutualism at least. And we see that they're both pretty strong and positive. In other words, the kids who start out high in vocabulary gain more on matrix reasoning ability, and the kids who start at high in matrix reasoning ability gain more in vocabulary. So these are the two parameters that represent the idea of mutualism at least. And then we can do various types of model surgery, so we can fix these parameters to 0 and see, does our model suffer? And yes, it does. In other words, we have considerable evidence here that you get this positive bivariate coupling between the two domains. Yeah?
Are these on consistent scales, and can we interpret the fact that the coupling seems to be stronger?
So this is a fully standardized parameter estimate to make it comparable. So yeah, these are kind of, this squared is the effect size, so it's about additional 4% of the variance, which is kind of the medium individual differences in effect size. So what does that mean, the mutualism? To summarize, there's an increase in matrix reasoning vocabulary, which we already knew. There were individual differences in growth, which we very much expected and would surprise no one, yet it's nice to have an actual parameter in your model that captures that. There's correlated change, even when we try to take into account of most of the dynamics. And we find that higher starting values are associated with lower rates of change within domains, so you get this dampening. But most excitingly, we find some evidence for mutualism or bivariate dynamic coupling. What does that mean? That a higher starting score on one domain is associated with greater gains in the other domain in this particular cohort. So, the summary or so what? I think this is interesting because it gives us at least some understanding of the positive manifold now. In my view, mutualism doesn't mean you can't also have a G factor, so I don't particularly believe that everyone starts out completely uncorrelated. There's too much genetic evidence that goes against that. But it does show, for instance, how things that might look entirely genetic could actually be caused by gene-environment interactions. So it gives us a bit of better understanding of cognitive development and also of cognitive challenges. So this is a really nice paper by Ferrer from a few years ago that did something similar, but then they looked at IQ and reading ability. So instead of matrix reasoning and vocabulary, they looked at general IQ and reading. So in the healthy population, you find this positive bivariate coupling between these two domains. In other words, if you read more, your IQ goes up a bit. If your IQ goes up a bit, you tend to start reading more. But they found in a subpopulation that suffered from dyslexia, they found no evidence of coupling. So it's not just the mean level of IQ or reading ability is different. It's the fact that the people with dyslexia, or at least the subgroup with dyslexia, didn't seem to benefit from reading in the same way as people without dyslexia did. And when their IQs went up, that didn't help them start to read more. And this could be interesting as an early warning system, so you could imagine something like this where you model dynamic change on a few time points, and the parameters you estimate allow you to extrapolate. So normally I'd say our school system is kind of geared towards some kind of cutoff, and then you're in trouble and you're not. But actually, if we have repeated measurements, we can have a far more complex picture of where everyone is going. So if you're a highly coupled system, your prediction over time is actually far better than someone who may be for kind of being born into a highly educated, wealthy family is doing okay now in terms of cutoff scores, but actually isn't developing in the same way we'd like them to develop. So how to SEM? How to fit these types of models? This is my brief intermezzo. Use R. I think you're going to do a lecture and workshop on this. So it's a very useful open source package that everyone can use, and you can fit the models I just explained. There's lots of really nice interactive tools online. You can make your research reproducible, you can make nice figures. So this is cycling in London. This is about 20 lines of R code, where you just grab a map of London, you grab the cycling data from the government, and you overlay them, and you get something that looks as gorgeous as that. It'll make you a better person. So I did an NH3 sample. It's a pair T test, and quite convincing results, I'd say here. So yeah, all go join this R course. If you want to fit the kinds of models I just explained, we recently posted a tutorial paper on developmental cognitive neuroscience using these latent change score models. And we include a whole bunch of R code that you can pretty much plug and play. So it's hopefully easy to use. Has a nice step-by-step analysis of how to do this kind of thing. And we also have an online tool which we made because we could. But the nice thing is here you can tweak these dynamic parameters to start to see what raw data would look like if these parameters were of a certain magnitude. Because I don't know about you, but I didn't initially have great intuitions about which dynamic parameters matter and why. But you can play around with these online and see what raw data would look like if your dynamic model was true. So after that infomercial.
Sorry, commercial.
Yeah. Back to
Where we get it?
It's on bioarchive. So it's freely downloadable. Currently under review, so it might be slaughtered and butchered beyond recognition, but it's currently still in this form. Which also means if there's anything that is unclear about it, please tell me so I can make the revision a bit better. And you can also approach me later, and I think they're going to post it online, so you should hopefully be able to find this. So yeah, go use R. So switching gears a little bit to the other end of the lifespan. So that was big data, or at least moderately large data, that I think because of the element of time forced me and my collaborators to think about development in a bit of a different way. So the project here, I'll focus on kind of later life aging in CAN-CAN, which I think is big data in another sense, namely the data richness, so the number of variables or the types of variables that are included, which allows you to go beyond what I would say is the more traditional literature. So what do I mean by that? Well, there's these three domains I'm quite interested in, which are fluid intelligence, processing speed, and white matter integrity, which have all been posited as either a symptom or a cause of cognitive aging across the lifespan. So there's lots of associations between all of these, and I won't walk you through all of them, but for instance, we know that declines in inspection time and reaction time are correlated with declines in fluid abilities in older people. So that's an example of fluid intelligence and processing speed being quite tightly coupled. We know that white matter integrity and processing speed are coupled. If you take a general summary of people's white matter integrity, it'll predict their processing speed, general information processing speed. And there's some work we did already in CAN-CAN, but there's many other studies that found that white matter integrity predicts fluid intelligence in old age. So I would say this kind of trifecta of bivariate associations is quite well established. If you want to look up any papers, there's a few here. There's literally hundreds on this, and the effect sizes are quite astronomical. But they're kind of generally all bivariate studies, right? They look at aging, they correlate these two things, and they either have some plausible causal account, but clearly there's something going on here. So I wanted to think about this a bit more and see if we can develop a model that accounts for all three of these interrelationships. So I went to the literature on psychopathology and stole a model from there, from Cannon and Keller, who proposed this quite nice model called the watershed model, and I'll walk you through what they meant by it. So they proposed it in the context of psychopathology. And they proposed it to explain the empirical findings related to schizophrenia. And so this is kind of a river, if you will, and 4 is the kind of outcome of the river. So the basic idea that I'll walk you through in a bit more detail is that there's a hierarchical main-to-one cascade of partially independent effects. What does that mean? It means that 4 is, say, the phenotypical outcome, like schizophrenia. And if we go one level up the river, there's these initial kind of components of schizophrenia, things like the higher end of phenotypes, such as working memory deficits and many other cognitive and emotional and behavioral challenges in schizophrenia. So these are kind of the high-level phenotype, like the endophenotypes. But we can go further up a river, I'll say working memory deficits, and look at slightly lower endophenotypes of biological correlates, such as dopaminergic regulation in the prefrontal cortex, which may in turn explain working memory deficits, which may in turn contribute to schizophrenia. And then finally, we have the kind of mythical genotypes, which are the ones that are almost invisible, And the idea is that these all together, via these intermediate stages, contribute to the phenotypical outcome of schizophrenia. So why is this a useful model? Because I think it explains or integrates various things we know to be the case, such as small genetic effects. So, for instance, for schizophrenia, I think till about 4 or 5 years ago, the total R squared for all genetic effects together for schizophrenia was on the order of 2, 3, 4%. And then recently they did it again in about 200,000 people, and that jumped to 12% R squared. Now, one potential explanation of that pattern is that there's actually lots and lots and lots of tiny effects. So only if you study 250,000 people will all of these tiny effects contribute to a larger effect or pass some statistical threshold. It also explains the lack of success of single cause explanations. So if the kind of overall system works a little bit like this, you aren't going to find the kind of gene for schizophrenia. So it doesn't work like Huntington's chorea, for instance. It also explains non-unitary consequence of lesions, so it's not always the case that if you do a single thing to a single person that you know exactly what's going to happen, because it depends on where that person is on a particular kind of arm, so to speak. But I think most importantly, it explains things like phenotypic heterogeneity. So if you look at the diagnosis for depression, which is, you could also use depression here, if you look at the DSM criteria, there's 9 symptoms which you have to display for at least 5 days for 2 weeks. At least 5 out of 9 for 2 weeks, and then you're depressed. So that means you can be depressed in 1,500 ways, just by the permutations of the symptoms profile, which is a nice factoid for our parties, but my colleague actually trawled through large data sets and actually found 1,000 out of those 1,500 potential types of ways of being depressed in actual people. So people really are depressed in 1,000 different ways. And in some sense it doesn't surprise us, or it shouldn't surprise us, but it also explains why if you're then going to link depression to the gene for depression, you're not going to do a great job, because you're trying to link small effects to 1,000 different things, and then you're disappointed if that doesn't work. So the way we wanted to adapt this model was to think about this same framework, but then the outcome isn't schizophrenia, but it's fluid intelligence. So what are the statistical implications if we take this idea of a riverbed seriously? Well, we should expect that the upstream measures, or the upstream variables, are more independent, potentially partially independent. So they have greater statistical dimensionality. So if they're partially independent rivers with partially independent genetic effects, then things get more and more complicated the further up I go. Secondly, there's this idea of multiple realizability of higher constructs. So you can have the same outcome, whether it's IQ or it's depression or schizophrenia, for different reasons. There's a many-to-one or a multiple cause at each level, so it doesn't just work if you take the biggest symptom and think you're done. You actually need to know all of them, so there should be this many-to-one prediction, and I'll show you what that looks like in a bit. And there should be hierarchical dependence. So if this really is this kind of causal system, that means for our purposes that fluid intelligence is caused or determined by processing speed, and processing speed is determined by biomatter integrity. It also means that there are no more direct relationships between these higher effects and these downstream effects. So we can map the whole system. We understand what causes what in what order. So we can translate these predictions into a testable SEM. I'll walk you through that. But first, a little bit about the sample. This is the Cambridge Center for Aging and Neuroscience, which started 2010, where we badgered 15,000 people in the Cambridge region via GPs to make it as population representative as possible. And almost 3,000 of those were convinced, cajoled into giving a home interview, which gave us an incredibly rich picture. And then about 650 or 700 came back and did an extensive battery of cognitive testing, resting state MRI, and MEG. But most importantly for our purposes, extensive structural MRI. And then 280 did task-based fMRI. But what you may or may not enjoy is that we made this entire data set publicly available through this CanCan data portal. So you can go here and see what we have, see if it aligns to your interest. And then you can apply for this data set, and then, assuming you're doing more or less legitimate research and you're willing to sign, I won't try to identify people, we can share this data with you. And 160 teams have done so before you, which our admin IT team loves, the fact that 160 people are downloading this type of data set. So what are the behavioral measures? So how are we going to translate this idea empirically in the context of Can-Can? So 550 did the Cattell Culture Fair test, which is the equivalent of a fluid test. We have 4 subtest scores, and we take the sum score on each of those subtests, which will be our index of fluid ability. Then we have 3 different RT tasks because we want to capture this idea that maybe there isn't one thing about processing speed. So we already know from the aging literature that things like your mean RT and your variance are quite different components, and they both predict mortality partially independently. So it's not just your top speed, so to speak, but also your stability or replicability of your own top speed that matters. And then finally we included this unqueued task where we didn't actually tell people to respond as quickly as they could, just to see whether that predicted unique variance. So for each of those 3 tasks we have the mean and the SD. So the simple RT is just when you see a light, press that finger. Choice RT is when you see a light, you have to press one of the other 3 fingers as quickly as possible. And then the final task, which was unqueued in the sense of trying to respond as quickly as you can, we just told people press a button if you hear or see anything on the screen. The neural measures, we use fractional anisotropy, which may or may not be familiar. It's a measure of white matter integrity, indirectly because we look at water dispersion. So the idea is normally water disperses like that, isotropically, just Brownian motion, but if it's constrained by a white matter tract, it'll disperse more frequently or more likely to disperse along the axis of the white matter tract. So we can quantify that as an index of fractional anisotropy, which varies between 0, which isn't great, and 1, which is good, hypothetically. So this Scottish study I talked about before, this is the actual white matter tract from one of their participants, which is amazing, and I won one for my birthday. So we looked at 10 tracts from this generic atlas. We took the mean of A for each of those tracts. So we can now go to empirically test the model, and we'll build it up in stages to kind of walk you along. So firstly, we have fluid intelligence, which is our outcome we're interested in. Well, you can see it doesn't fit the single factor model, and in our case, it does. Green is good. So it seems as though we can summarize performance on these 4 subtests as a single factor. So now we go into processing speed. So remember, we have these 6 different factors. So these are our intermediate endophenotypes, if you will. So firstly, we would expect, based on the watershed model, that we can't summarize all of that into a single number. So we can test that. But a single factor where you just have a processing speed factor doesn't fit at all. So it fits terribly, and also various alternatives such as have one factor for speed and another for consistency also didn't work. So it really seems as though there is this kind of complexity in here that we can't easily reduce to 1, 2, or even 3 numbers, which is in line with what we expect on the basis of this watershed model. So we can take those 2 steps together, processing speed and fluid intelligence, and we can fit what's called a mimic model, where we have multiple indicators, so multiple measures of fluid intelligence, and then multiple causes. Namely, all of these different aspects of processing speed should matter a little bit to the end product of fluid intelligence. Now this is exactly what we found, so A, the data fit rather well. And then we can see here, out of our 6 measures of processing speed, actually 5 make unique contributions to fluid intelligence. So this is a bit like a multiple regression model. So each of those 5 contribute unique variance in explaining what is, I think, a surprisingly large proportion of the variance. So 58% of the variance of fluid intelligence we can predict by just these processing speed measures. And you can see that even if you take, say, the choice, reaction time, speed, after that you still have quite a few pretty chunky parameter strengths there that determine fluid intelligence above and beyond your mean performance on this choice RT task. So far so good. Seems to be in line with the idea of the watershed model. So how about white matter integrity? Well, again, we can see, does this fit the single factor model? It doesn't. And we tried various kind of exploratory factor analysis. You need 4, 5, or even 6 factors to explain the covariance among these tracks. So it isn't the case that you have a good white matter brain or a bad matter brain. There's more going on. This is true even if you regress out age. So it's not just the fact that some tracks age differently than others. So our interim summary of our data so far is that fluid intelligence, the 4 fits a single factor model. Processing speed is multidimensional, so we need more than 1 factor. this processing speed predicts fluid intelligence in a many-to-one fashion. In other words, 5 out of the 6 measures make unique predictions of fluid intelligence, and white matter is multidimensional. So we can now put all of it together in one big model, which looks like this. Don't be scared. This is one of the famous psychometricians called the butterfly net model, because you can just pick it up and start catching butterflies with all your parameters. So, to walk you through it, firstly, the model fit. Okay, that's a necessary but not a sufficient condition. It means we can start looking at the parameters. Again, we see that, very similar to before, 5 out of the 6 processing speed measures predict variance in fluid intelligence. But then, interestingly, we see something very similar going on with the white matter tracks. So we see that there's definitely a white matter track, so the forceps minor, which are the kind of connecting the two frontal lobes, gives you the strongest parameter estimates, but above and beyond that, lots of other tracks, including the corticospinal tracks, the SLF, make unique contributions in unique measures of these outcomes. So this many-to-one idea that's captured by the watershed model is preserved here.
Excuse me.
Yep.
Does this model assume that the white matter measurements only contribute to fluid intelligence via the
Yes.
speed?
Yes, exactly.
Is that a testable function?
So that's in there. So if these made direct predictions to fluid intelligence above and beyond what's captured indirectly, then your model fit should be poor. So essentially any arrow you don't see, and there's a lot, but there's also 60 that aren't here, if the path would be significant or strong, it would contribute to overall misfit of the model. So there's one exception, which actually is the forceps minor, which if you allow it to directly contribute to fluid intelligence, it does improve model fit. So it fits more than well enough like this, but there is something residual we're not capturing by this model, which is unsurprising because this is only a small subset of it. So we can then compare model alternatives, right? So this model fits okay, you get a bunch of significant parameters, but what else could we posit that maybe it's just a generic feature of a covariance matrix that everything correlates with everything, so all of your models would fit okay. Well, we could invert the layers. We could say, well, maybe everything's correlated here, so if I invert this and this layer, it does almost equally well. So we tried this, but this model fits much more poorly. In other words, there seems to be far more strong residual correlations between the processing speed measure and fluid intelligence than vice versa. Now, a criticism you might have of this model is that, well, this inverts not just the causal order, but there's now 10 here and 6 there. So we did all permutations that would give you 6 of each at each layer, and then compared all of them in the watershed model, the original representation, again dating my age quite considerably, wins every time. So what about other alternatives? Well, you could say, well, maybe there is some difference in the white matter tracks, but maybe some tracks are more important than others, but their contribution is equal for all dimensions of processing speed. So this you can test in SCM by what's called equality constraints. So you tell your model, you can freely estimate this parameter, but it has to be the same for, say, this singular tract for each of the outcomes. And it has to be the same for the forceps minor to each of the outcomes. So of course you save yourself quite a few parameters here, so your model is far simpler. But the question is, does it do equally well? And in our case, it didn't seem to be. So even though we saved 40 parameters, the model where you estimate each of them individually still outperformed this model. And now a final alternative, so you say, well, maybe there are differences between the dimensions of processing speed, but they're then equally determined by each of the tracks by some generic feature of your white matter integrity. So again, we're going to save a few parameters by equating whatever the prediction to the choice reaction time mean speed is, but it has to be equal for each of the tracks. And this model, again, fits considerably worse despite saving ourselves 54 parameters. So we can then say, well in CAN we have white matter, cognition, 2 levels of cognition. But what would the genetic prediction be? We do have spit in a cup, but we're not sure what to do with it just yet, but they're collaborating with various teams. Arguably 600 would definitely not be large enough for these types of questions, but we can see what predictions does the watershed model make about genetics. Well, if we think about the kind of genetic architecture, we would expect that there is this shared prediction. So whatever genes affect white matter should also affect processing speed, if this idea holds. This is a very nice paper by Kochanov that was published when my paper or our paper was in revision or just rejected, which made me really annoyed we didn't post a preprint, because it would have been a prediction if we posted a preprint. Now it's a postdiction. But it was a prediction. Namely, we would predict shared genetic antecedents for white matter and presence speed. So it's a very nice study in 2 different twin samples where they saw there's a significant degree to which common genes affect both variation in FA and brain presence speed. So this is exactly what we'd expect if the watershed model is true, or at least is an accurate representation of part of the data. So to summarize, the data is compatible with the watershed model. Both presence speed and white matter integrity are not unidimensional. There's a many-to-one mapping at both of these levels. There's many-to-one from presence speed to fluid intelligence, and many-to-one from white matter integrity to presence speed. The inverted model is significantly worse, and there's some preliminary evidence for the genetic predictions we would expect to be the case. So I would say this is a plausible account of inter-individual differences in age-related changes in fluid intelligence. Of course, longitudinal data would be the next step to test this truly. Okay, finally, a very brief toe dip into big data, capitalized. So recently, finally got access to the Biobank, which is an incredible public resource if you don't know about it already. So essentially, they linked data from the NHS to a subset of people who came back and did imaging and did cognitive testing for a lot of people. And there's a really nice paper by your group, by Carla Miller, who looked at the genetics and the imaging data for a first pass. So what do I mean by a lot of data? Or how does it work? Well, you can go to their website, which works incredibly well for a project of this size, and you can say browse, which all sounds quite normal, and then you can go to the UK Biobank Assessment Centre, where you say, well, I want to look at the cognitive tasks, and say I'm interested in the cognitive tasks of presence speed. And this still looks reasonably normal until you look at that. So there's 500,000 people waiting for you who did a presence speed task, and 8 other tasks. And then there's this another innocent tab which says, oh, by the way, we have 3 waves of 10,000 of these people. It's absolutely astonishing. And it's only recently that people are digging into this in a bit more detail. So what are the challenges I've encountered over my 5 days working with this data in the run-up to this talk? I mean, big. So I have a fixed 16 or 15-inch MacBook Pro, which starts to hum loudly whenever I do anything or go near the Biobank. So even in R, I needed a new package specifically to read in the data. So if you just say read in my data, it collapses. So it actually has to do a special procedure where you read in chunks of the data and then bolt them back together again. Otherwise, no dice. So this is 500,000 people on 4,000 variables. There's 2 billion cells. It's big data. I'll show you a figure that took about 3 minutes to render on my MacBook Pro. I tried to make a longitudinal spline plot so to fit a nonlinear curve. It didn't work. It said go away. And things like significance are meaningless. So I recently learned you just worked on base factors last edition. So to give you an idea of the base factor for the linear model, this is p is smaller than 0.005. And so for reference, there are 10 to the 82nd particles in the known universe. So this is pretty strong association. There's also the flip side of big data, which means that you often don't get exactly the test you want, which for certain things doesn't matter, but also can affect the data quality. So the fluid intelligence, which I'm particularly interested in, on first glance doesn't seem to be a particularly robust test. So it doesn't capture fluid intelligence in anything like the same quality as the Cattell does. So that's the kind of balance you need to take, although the presence speed measure seems a bit better. So often, in return for 500,000 people, they're not going to spend 12 hours on your favorite task. So sometimes you get lower quality data on some cognitive tasks, although the imaging data is quite nice. You can ask for the raw data, by the way. 10,000 DICOMs, if you want to annoy your IT colleagues. So just to show you very quickly what it looks like, relating presence speed to age, looks like this. And you might ask, why didn't you draw a confidence interval? I did. It's there. So this is 500,000 people. And a linear model, because as I said, a spline didn't render properly. This is white matter tracks. So the biobank is about age 40 to 70. The idea is that they will also follow these people longitudinally. So currently they already have 10,000 people imaging data. So there's 10,000 people, brain matter, white matter, resting state. You can ask for the raw data or you can ask for nice extracted data. So yeah, go use it, it's amazing. So to summarize, big data is very promising for developmental neuropsychology because it allows you to take a more of a systems approach to neurocognitivation. So not just look at the question you're particularly interested in, but also look at what might matter for the questions you're interested in. It can develop and inspire new theories, so it forces you to think about your favorite topics in a different way. And you can test theories as models. Challenges, several, but opportunities are almost endless. Thanks to the funders, the collaborators, and the mascots. Thank you very much.